Dear Professor Keck, thanks for being with us. The floor is yours.
Thank you so much. Good morning, Mexico. I'm pleased and honored to be able to give you that webinar, which we call Staying Mentally Healthy in Stressful Times, as Andreas Schötz already mentioned. We indeed actually do live in stressful times. I think we are in the midst of the second wave of the so-called corona pandemic. But what are the consequences? People do suffer from a lot of things, but what is the psychological, the mental health point of view? Well, to shed light on this phenomenon called stress, during the corona pandemic, researchers from the University of Basel, the oldest university, by the way, in Switzerland, conducted during both waves, also right now, of the pandemic, an online assessment and interviewed about 10,000 people. And what they found out is the following. A lot of people are stressed out. They saw a 40% increase in perceived stress. They saw a nearly up to 60% increase in depressive symptoms, and they found a 20% increase in sleep disturbances. So huge differences, huge disturbances. What are the consequences of stress or increased stress? What in this context means stress difficult to cope with. So the stress hormone regulation is deeply deranged. We can define stress-related disorders as diseases with etiology, onset, development, which is substantially influenced by stress and its neurobiological correlate. And there's a huge variety of stress-related disorders. There are psychophysiological disorders like ulcer or asthma. There are cardiovascular disorders such as angina or hypertension. We know immunologic disorders, but most important, we see a lot of mental disorders such as depression, PTSD, of course, and sleep disorders. I would like to share some case reports with you illustrating mental health problems in daily practice. I'm sure patients that you all know very well. So the first case is a 45-year-old female. She's a receptionist, 3 children. Husband is actually unemployed due to the pandemic. She suffers from sleep disorder since about 6 weeks, and she is only able to sleep 2 to 3 hours per night. She's suffering from dizziness, exhaustion, loss of concentration, rumination, and she cries a lot. So we could try, just to give you a few hints, we could try sleep hygiene management, stress management, we could do couple therapy, but we need an urgent action. We need a solution right now. So should we give a benzodiazepine, analogon like Zopiclone, by the way, or benzodiazepines? Well, whatever we administer, we have to be aware of the side effects and of drug-drug interactions, that is pharmacodynamic interactions. And the next case gets closer to that. It's a 78-year-old male, a former lawyer. He's widowed since 5 years, one of my patients. He suffers from sleeping disorders since about 3 months. He's suffering from daytime sleepiness. He has cardiovascular disorder. He suffers from hypertension and diabetes. And due to that, he has to take about 5 drugs a day. So quite a lot. And well, we could give benzodiazepines, but I already mentioned pharmacodynamic, so-called drug-drug interactions, which is a preventable cause of a lot of morbidity and mortality. Benzodiazepines, of course, I'm sure you're familiar with, are effective, no doubt about that, but double-edged, especially in the elderly. They cause tolerance and dependency, they cause dizziness, drowsiness, muscle weakness, what altogether then increases the risk of falls and cognitive disturbances, to name just a few. So we should be reluctant in administering benzodiazepines. I'll come back to that. The next important point is, how do we sleep? What is sleep? Sleep can be characterized via EEG waves and consists of 5 stages as depicted here. From light sleep to deep sleep, it goes down a stage, down the stairs, from light sleep to deep sleep and REM sleep, where we preferably dream. It consists of cycles which need to have a specific and, this is important, individual architecture or shape to ensure that sleep is restful and recreative. A disruption is harmful. Deep sleep during the first half of sleep. You can see it on the left-hand side of the diagram. So the deep sleep comes about 90 minutes after we fell asleep. And the REM sleep, the dream sleep, occurs later. You can see the dark blue and the light blue colored squares. The longer we sleep, the more REM sleep we get. If sleep is disrupted or does not have your individual or specific shape or architecture, it will not be recreative. In all psychiatric disorders, sleep disturbances and the non-restful sleep are very common. It's the rule rather than the exception, as you all know. Just to give you a quick overview, when focusing on affective disorders, you can see nearly all patients are suffering from current awakenings, that is a disruption of sleep continuity. They have a reduction in deep sleep, that is the stages S3 and S4, as I showed you. They have a decrease in REM sleep latency, so REM sleep occurs too fast and too often, and they all suffer from hypersomnia. Why do we sleep? To be quite honest, we do not know precisely, but we know that sleep is regulated via two processes or factors. Adenosine on the upper panel accumulates during daytime or during being awake. So it accumulates and accumulates and accumulates, and it creates at the end of the day what we call sleep pressure. So at the end of the day, we are overwhelmed by sleep. We can't resist. The second process factor is melatonin, what is secreted from the epiphysis or pineal gland. It's the internal clock or timer. It's a very important circadian factor. We call it the biological clock. When it gets dark, it's released out of a sudden and it brings you to bed. Adenosine brings you also to bed, and melatonin switches off the light, as we can circumscribe it. What is of interest, blue light, as emitted from smartphones or LED displays, inhibits melatonin release. So be reluctant using smartphones or LED displays when going to bed or in your bed. So that is what we call the physiological control of sleep, the two-factor or two-process model, the homeostatic factor adenosine, and the circadian factor, the biological clock, what is well known as melatonin. There are certain, and we know them very well, demographic risk factors for insomnia. Women and elderly most often suffer from insomnia. There's an increase of up to 50% during menopause or menstruation. So insomnia is more prevalent in women, especially when being in the menopause. In the elderly, they are disproportionately affected by insomnia. They often have what we call a light sleep, a disrupted sleep. And what is also very important to mention, insomnia or disturbed sleep or a non-restful sleep is both a risk factor and an early sign of depression. So it's very important to ask your patients, how is your sleep? I already told you, apart from emergencies or very critical situations where benzodiazepines do a very good job, is there an alternative to benzodiazepines in treating insomnia, as in the two cases I showed you before, where we should be reluctant to administer benzodiazepines? Of course, there are alternatives. There are antidepressants, certain antidepressants, which regulate in a very beautiful way your sleep architecture, such as trazodone. But there are also, and this is even better, plant-based traditional approaches, such as valerian and hops. Hops are the flowers of the hop plant, a flavoring and stability agent in beer, what is very well known in Germany and I'm sure in Mexico also. And well known in herbal medicine or traditional medicine is valerian root as medicinal herb. So what is very promising is that Seti 91019, so the combination of hop and valerian, mimics the very natural way of sleep regulation I just showed you before. That is the two-factor or two processes model. Valerian, as depicted on the left-hand side, acts as an adenosine receptor agonist. where caffeine, by the way, acts as an antagonist to keep us awake during daytime. So adenosine mediates action on such receptors that are also addressed by valerian components. Hop, in turn, acts as melatonin receptor agonist, so regulating and inducing sleep in a very gentle way, these two together. And we also have what is promising data, in vitro data, suggesting that the valerian component of Seti 91019 has also an agonist effect on the GABA receptor system, what is the well-known mechanism of benzodiazepines. And I'll come back to that in a minute. With regard to sleep latency, that is the time to fall asleep, the combination of hops and valerian is superior to valerian monotherapy and of course superior to placebo as depicted in this diagram from the study of Kötterer et al. Needless to mention that such a traditional or herbal medicine has no adverse effects. There is a lot of clinical evidence. Just to quickly show you the literature documenting the effectiveness, it has a proven clinical efficacy. It has an excellent safety and tolerability, what is known since ages. It improves, as I showed you, sleep architecture. It reduces nightly awakenings and increases sleep duration, what is very important in old age patients. And it reduces sleep onset, that is sleep latency, over placebo and valerian mono extract. Very nice effects. And there's a beautiful improvement of the main sleep parameters. A study from Fissel et al. Published in 2000. Seti 91019, that is valerian and hops together, reduce sleep latency as well as the total awake time per night. On the left-hand panel, total time of deep sleep is increased, very important for recreational sleep, for that kind of sleep quality. And patients felt more refreshed in the morning. Here are two very beautiful hypnograms of the EEG wave sleep architecture. On the left-hand side, you can see a hypnogram of a chronic primary insomnia patient. A patient suffering from chronic insomnia, and it looks quite horrible. You can see very frequent nocturnal short arousals at the dark black colors. You can see a few fragmentic REM phases where we sleep, and there's practically no deep sleep phases during phase S3 and S4, though that sleep is very bad. It looks horrible. And on the right-hand side, you can see the effects of only 2 weeks treatment with Seti 91019, valerian and hops. Sleep architecture is mainly restored. It looks very sorted out. And the REM latency, by the way, is still shortened. You can see it on the very left side. REM sleep occurs shortly after fallen asleep, what is a typical sign of depression. But we see distant REM phases, and we see definitely more deep sleep phases and less frequent nocturnal awakenings or arousals. To cut the long story short, it's efficacious and safe. We have no risk of dependency, for example, and it's very well tolerated by patients. So the use or misuse of benzodiazepines is a huge problem, and we should prevent it whenever and wherever we can. But what if it's already too late? Flesch and colleagues in that study published in 1997, they conducted a small but very interesting study. Over a period of 4 weeks, they administered 2 tablets, that is about 500 milligrams, valerian and hops, to see potential changes in hypnograms. And on the left-hand side, apart from the other ugly side effects, you can see it again. Benzos decrease S3 and S4 deep sleep. Sleep is not recreative or restful. This corresponds with our clinical experience. It brings patients to sleep, but it is flat and rather useless sleep. You can see just a few REM phases on the right-hand side of the diagram. Patient often is awake. There is absolutely no recovery after sleep. And this is what happened on the right-hand side after a 4 weeks treatment with Seti 91019. You can see Again, a clear separation of sleep stages. Sleep stages are longer lasting. There are more REM phases. Patient is not awoken, so there was a good recovery after sleep, a normalized, a totally, I might say, normalized sleep architecture after the disruption of benzodiazepines, and a 4 weeks treatment with hops and valerian. Quite impressive. We do not see any drug-drug interactions. I already mentioned that very harmful pharmacodynamic interactions, and we couldn't see it even in the combination with hops and valerian, no clinically relevant interactions either. So it's very beneficial for patients that are already on several medications, like the 75-year-old gentleman I showed you before. Another case report leading us to another huge problem in mental health. It's a 55-year-old female. She's counselor. She has 2 adult children, and she's divorced since a few years. She is suffering from sleep disorders since about 8 weeks. 2 to 3 hours sleep per night only. Sleep is disrupted. She is suffering from exhaustion, a loss of concentration, rumination, cries a lot. She has already ongoing, according to the guidelines, cognitive behavioral therapy, what is very effective. She is doing sleep hygiene management after teaching. She is doing stress management, and she already takes benzodiazepines, temazepam in that case. So she already is on benzodiazepines, and she is still suffering despite doing a correct and guideline-oriented cognitive behavioral therapy, including sleep hygiene and stress management. She's still suffering from rather severe depression. So there is a definite need for add-on therapy, so medication. But what shall we give? She is, I forgot to mention, in the midst of her menopause. Sleep disorders are very common during menopause, as I showed you, an increase of up to 50%. And during menopause, there's also a 3-fold increased risk for depression and a lot of bodily symptoms, as we all know. So what does the World Health Organization says? Depression and other stress-related disorders, such as anxiety disorders, will be on number 1 in the ranking of causes of death or chronic disability, the so-called disability adjusted life years, the DALY years worldwide. Though this is very impressed, stress-related disorders as depression are heading coronary heart disease, accidents. I just recognize this is still in German. I'm sorry, but the rest is not that important. Number 1 is depression. And by the way, about 350 million people worldwide are affected right now, and the highest prevalence of 6%, whereas 5 to 4% in the rest of the world are in the Americas. What is very interesting. Depression is a chronic disease. These are data from CAPFA from the early 90s of the last century. And if you have a first episode of depression, there's a risk of 50% to get the second. After the second episode, you will have a probability of 80% to get the third. And after the third episode, it's almost 100% to get it again. So there's a definite need for long-term treatment. And that is what we have to keep in mind. Long-term treatment means long-term medication in most cases. When treating depression, when doing psychotherapy and pharmacotherapy, you always should combine that. There is still considerable room for improvement, as we all know. About 40% of patients do not or hardly respond to treatment. 5 to 10 patients even deteriorate. And there are high relapse rates, 50 to 80% after recovering from an initial episode. There is a relapse rate about 54% after 2 years and about 80% even after 5 years. So this is really disappointing news. There's a special situation when it comes to geriatric depression. What is geriatric depression? It's depression after the age of 60. And in the case of geriatric depression, 50% of patients suffer from at least 3 and 20% from 5 or more comorbidities, like the gentleman I showed you before, cardiovascular disorders, hypertension, diabetes, and so on. Especially when following guidelines, there's a high risk, an increase in risk of polypharmacy. And polypharmacy in that context means 5 or more drugs per day. And there is a very nice study in Switzerland showing that people in nursing homes take up to 13 drugs a day. The maximum was, to just mention that, 27, what replaces a meal. St. John's wort as a traditional medicine, as a herbal medicine, today is an established antidepressant. I just quickly guide you through the evidence. There's a Cochrane study, including 29 studies published in 2008, clearly showing that it is superior to placebo in patients with major depression, that it is similarly effective as standard antidepressants, and most important, I feel, it has fewer side effects than standard antidepressants in mild to moderate depression. And all guidelines, I might say worldwide, I'm happy that I was part of both teams, developing the Swiss guidelines. All guidelines recommend St. John's wort as a first-line treatment when it comes to mild to moderate depression. Does it work? What about CE 117? Of course it works. There's a quick overview of clinical evidence, proven clinical efficacy. It reduces Hamilton depression, the common rating scale when testing antidepressant drugs. The Hamilton depression score more than 40%. There is a clear non-inferiority to fluoxetine, a common SSRI, and imipramine, the classic tricyclic agent, the first antidepressant. There is a very, very low drug-drug interaction potential, pharmacodynamic potential, and there are less, 60%, so to say, 60% fewer unwanted side effects compared to fluoxetine. And what are the common side effects when we use SSRIs? It's dizziness, it's agitations, it's gastrointestinal disturbances such as diarrhea, it's nervousness, and very important, sexual dysfunction. Schrader et al. Published a nice study in 2000, clearly showing that Seti 117 is non-inferior to fluoxetine, a common SSRI. And Bradström and colleagues were able to show that there's also a very nice long-term safety. They administered it over a period of 52 weeks, and it was safe and efficacious. It further reduced, that is also important, the symptoms of mild to moderate depression after a treatment period of 26 weeks. What is very nice results. Since I already mentioned it, since the serendipitous discovery of the first antidepressant imipramine in 1957 in Switzerland, it has been thought that its clinical efficacy relies on the mode of action of reuptake inhibition of biogenic amines, that is serotonin, noradrenaline, or dopamine. So it was all about monoamines in the synaptic cleft and the presynaptic neuron, because it was found in animal experiments that there is an increase in neurotransmitter concentration in the synaptic cleft after administration of an SSRI. We know today that this is most likely a sort of an epiphenomenon because the molecular effects are more downstream and took longer. And that is why we are very happy that by Kexel and colleagues and published in Phytomedicine in 2019, there is a very promising a new mode of antidepressant action of St. John's wort. It has an impact on cell membranes of the nerve cells. When having chronic stress, and it's also chronic stress that I refer to, there's an increased cortisol production. I already showed you that it is well known that in major depression, the brain loses control over stress hormone regulation, and there's an increase in stress hormones such as cortisol. As a result, brain neurotransmission is deeply disturbed, which can then lead to a disturbed regulation of emotions, cognition, and so on. You got stuck in a rut. You can't modulate your emotions any longer. And during chronic stress, there's an increased fluidity of the nervous cell membrane. So structure is changed and the concentration of the very important membrane proteins. That leads in turn to changes in receptor function, which are all located in the nervous cell membrane, that B layer. Cell membrane receptor function, enzymes, transporters, and ion channels are disrupted in their function. And then in turn, it comes to the end product, the change in neurotransmitter systems. So those biogenic amines, those monoamines, such as serotonin, dopamine, and adrenaline, leading at the end of the day to depression. And the interesting thing, when administering St. John's wort, when administering Seti 117, the fluid, the stressed cell membrane turns back to normal. Seti 117 normalized the cell membranes of stressed cells, nervous cells in the brain. So this is a very interesting and very new potential mode of action, the impact on nervous cell membranes. There unfortunately is one big problem with St. John's wort, its potential for pharmacodynamic interactions. For a long period of time, hyperforin, one component of the multi-substance product St. John's wort, hyperforin was thought to be the antidepressant agent in the herbal thing, and therefore some pharmaceutical companies even tried hard to produce drugs or preparation rich in hyperforin. But today we know hyperforin in reality is the bad guy, because it's responsible, it's the constituent responsible in St. John's wort that causes drug-drug interaction, because it activates, among others, the liver enzyme family CYP3A4 that is responsible for a lot of drug metabolism processes. And a systematic meta-analysis of 31 prospective clinical trials could clearly show that all studies using hyperforin-rich extracts of St. John's wort showed a CYP3A4 induction, and none of the studies with hyperforin-low extracts showed evidence of relevant interactions. So, and there is good news. Seti 117 has the lowest hyperforin, as you can see on the left-hand side left-hand-sided bars of that diagram. It has the lowest hyperforin concentration of all drugs or St. John's wort preparations available. That is less than 1 milligram hyperforin per day. And we know then there are no significant interactions. So Seti 117, the product with the lowest hyperforin concentration, and European Medicines Agency, EMA, and the Herbal Medicinal Product Committee stated that a daily dose of less than 1 milligram hyperforin does not cause any clinically relevant interactions. And there's a most beautiful study conducted by Catherine Sana in 2019, a cocktail study on interaction specifically addressing Seti 117, a drug cocktail containing 7 different medications, what is the so-called Geneva cocktail, non-alcoholic, I'm sorry, but instead containing 7 drugs such as omeprazole, midazolam, caffeine, bupropion, which are very well known to cause interactions which are metabolized by CYP3A4 enzymes and the very important transporter PGP, P-glycoprotein. So this cocktail is very well established to detect drug interactions. And what she found is the following: there was absolutely no induction of drug metabolizing enzymes, no induction of cytochrome P450 or PGP transporters could be found. So Seti 117 contains, as I already mentioned, a very low level of hyperforin and therefore has a low risk of interaction compared to other St. John's wort preparations. Of course, there's one crucial and scary question. What if Hyperforin would, at least in part, be responsible for the antidepressant, the clinical antidepressant effects of St. John's wort. And we are lucky to have that study from Lagman et al. Already published in 1998, clearly showing that there is no dose dependency of hyperforin with the efficacy of St. John's wort. Showing in the lilac color a preparation with 5% hyperforin, and showing in the light gray bluish color Seti 117, a very low concentration of hyperforin. There is no clinically relevant difference after 42 days of treatment in depressed patient. There is a difference of 1.1 Hamilton Depression Scale points, what is completely of no relevance. So hyperforin content, we can conclude, does not determine the clinical efficacy of St. John's wort. Lucky we are. So the efficacy of St. John's wort is not hyperforin dependent or dose dependent, as depicted on that beautiful slide, showing that increasing the dosage from the left to the right-hand side, increasing the dosage of St. John's wort does not lead to greater efficacy. And Seti 117 contains 500 milligrams, so on the leftish side of the diagram is on the safe side. There is no dose-dependent change in antidepressant effect as depicted here in percent change of points on the Hamilton Depression Scale in the dose range of various St. John's wort extracts. And it ranged from 180 to 1.8 grams, what were examined. And what is, from our clinical point of view, of uttermost importance, St. John's wort, Seti 117 has a clearly better safety profile than SSRIs. Shown here fluoxetine and paroxetine, citalopram, very well known and often used SSRIs, and sertraline, of course, fluvoxamine, a classic one, and the hypericum extract Seti 117 on the right-hand side. Nearly no, we might say, side effects, what could disturb our clinical treatments. And the standard today, of course, are SSRIs and not tricyclics. So, to conclude, plant-based drugs, herbal drugs such as Seti 117, St. John's wort, or Seti 91019, valerian and hops are effective. They work, no doubt about it. There are minimal drug-drug interactions, so no induction, as I showed you, of drug metabolizing enzymes or transporter proteins, SP, glycoprotein, and they are safe. They are safe, what is very important for our older patients, for our patients suffering from several comorbidities, for the elderly, and it's safe to give during menopause. Thank you so much for listening. I hope it wasn't too quick. I hope you could follow my German English, and I'm happy to answer your questions.